What's going on, guys? So today I want to talk to you guys about bacterial DNA replication. But before I do that, I want to talk to you guys about the structure and function of DNA. DNA functions as the basic unit of heredity. It contains 3 subunits: a phosphate group, a deoxyribose sugar, and a nitrogenous base. The nitrogenous bases include adenine, thymine, guanine, and cytosine. The nitrogenous base subunits pair to form this little guy. This is DNA. And like I was saying before, this is the basic unit of heredity. And if we go over here, we can see what it looks like in a bacterium. You can see that it is circular, but still retains its anti-parallel shape. DNA replication involves a multitude of steps. But where does it start? In the bacterium genome, there is a location known as ORI, and that just basically means origin of replication. There is only one origin of replication in the bacterium genome. The first part of replication involves the binding of DNA helicase to the ORI in the 3' to 5' orientation. As DNA helicase unwinds DNA, a region known as the replication fork is produced. This region promotes replication by enlisting a multitude of enzymes. The replication fork involves 2 strands of single-stranded DNA. The leading strand, oriented in the 5' to 3' orientation, is synthesized continuously throughout DNA replication. The lagging strand, oriented in the 3' to 5' orientation, cannot be synthesized continuously like the leading strand. Before I want to go further into DNA replication, I want to talk about the individual enzymes responsible for synthesizing new DNA strands. DNA polymerase III is responsible for DNA synthesis in the 5' to 3' orientation. The beta clamps attach to and support DNA polymerase throughout the process of synthesis. DNA gyrase, located here, prevents supercoiling of the DNA. Single-stranded binding proteins bind to single-stranded DNA to provide some structural support. Because DNA polymerase III can only synthesize new DNA strands in the 5' to 3' orientation, the lagging strand undergoes a different procedure. This procedure involves the recruitment of 2 new enzymes, DNA primase and DNA ligase. Primase is responsible for synthesizing RNA in the 5' to 3' orientation. The RNA primer acts as an expendable template so that DNA polymerase III can synthesize in the opposite direction. Synthesis of the lagging strand produce Okazaki fragments. These fragments are then bound together by DNA ligase. Although there's only one replication fork, DNA replication occurs in both directions. So whatever you see here is also happening over here simultaneously. Because bacterial DNA is circular, a problem occurs at the end of DNA replication. At the end of DNA replication, the 2 strands become supercoiled. In order to eliminate the problem of supercoiling, DNA gyrase cleaves, then rebinds respective DNA. And that is the process of DNA replication in a bacteria. Thank you guys for watching.